A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 3, Mass Spectrometry-Based Proteomics. Lecture 4, Peptidomics.
Good morning, ladies and gentlemen. It is an honor to host distinguished proteomics scientist Professor Anthony Purcell, who joins us from Monash Biomedicine Discovery Institute, Australia. He is one of the most cited Australian proteomics researcher and a leader in immunopeptidomics with over 230 publications. Professor Anthony is Principal Research Fellow of Australia's National Health and Medical Research Council and Deputy Head Research of the Department of Biochemistry at Monash University. His laboratory focuses on how the diverse array of peptides presented to the immune system, known as the immunopeptidome, is influenced by infection, inflammation, and the environment. He has made significant contribution to understand the role of antigen presentation, including the characterization of T-cell epitope in several autoimmune diseases. Also, he has studied drug hypersensitivity, cancer, and infectious diseases. He is well known for the work that has highlighted the role of post-translationally modified antigens in immunity. He has been instrumental in bringing new quantitative tools to immunological studies, particularly the use of multiple reaction monitoring to calculate the cell surface expression levels of specific HLA peptide complexes. Professor Anthony is Australasian Proteomics Society's Vice President and is on the executive committee of several other learned societies. He is also the council member of Human Proteome Organization. Professor Anthony also serves as scientific advisory board member of Human Proteome Project. He is an executive advisory board member of the journal Proteomics, an associate editor of the journal Molecular Immunology, and an editorial board member of Molecular and Cellular Proteomics. He consults widely for industry and is on the scientific advisory board of Evaxion APS, an immuno-oncology biotech startup in Denmark. Now, we would like to invite Professor Anthony Purcell for sharing his lecture. Thank you.
So, I guess in the next 40 minutes or so, I thought I would talk to you about a subject that's very dear to my heart, which is immunopeptidomics. And as you've heard by that very thorough introduction, I hope I'll live up to everything. This is the study of peptides that are involved in immune recognition. And I'll focus my discussion on cancer-specific peptides that are potential T-cell epitopes in a variety of different cancers. So I thought I'd just start off by defining what the immunopeptidome is. And its genesis. And what I'd like you to think about as I discuss this is really what the immune system's been doing for many hundreds of thousands of years is essentially what we do in proteomics, and that is taking protein level information and translating it into peptide level detection. So when we think about cancer in particular, there are changes in the proteome, as we know, and the immune system is able to sense this by taking endogenous proteins, including proteins that may be specific to the cancer, aberrantly expressed in the cancer, and part of the normal protein degradation in the proteasome yields peptides that sort of get hijacked by the immune system. These short peptides get transported into the endoplasmic reticulum, where they're able to be bound to MHC class I molecules, human leukocyte antigen class 1 molecules in humans, there's a series of chaperones that are involved in optimizing this binding. Then they egress to the cell surface presenting their peptide cargo. So you can imagine when you're thinking of all the proteins in the cell, this is quite a complex mixture of peptides bound to the HLA molecules. And for some of these peptides, there will be a T cell, in this case, a killer T cell, that is able to recognize specific tumor antigens and elicit immune response. And, you know, I guess in a perfect world, we'd all have T cells that recognize tumor antigens, and those T cells would be able to go on and kill the tumor cell before it caused problems. So today I'll discuss how we go about studying these peptides, some of the types of peptides that we're particularly interested in, and in the latter half of the talk, I'd really like to focus on really taking what has been quite fundamental studies in cell lines and moving into more clinical samples and some really exciting work I think that we've been developing to sort of move into more of a translational phase of our research. So today we're going to focus exclusively on this aspect of the immunopeptidome, the class 1 pathway. There's a second pathway in professional antigen presenting cells that many of you may be familiar with, and this involves the uptake of exogenous antigen in phagocytes, etc., and processing of these exogenous antigens, binding to MHC class 2 molecules and presentation to CD4 positive T cells, or so-called T helper cells. And these might be involved, for example, in eliciting a humoral or antibody response. The other thing I'd like to just sort of introduce to you is that everyone listening to this presentation will have a different complement of HLA molecules, and indeed HLA class 1 molecules. And this is because this region of the genome is extremely polymorphic. And as you can see here for the 3 HLA loci, there are a large number of different allotypes which encode for distinct proteins. So over 6,000 proteins, and this is probably quite just at the tip of the iceberg in terms of the diversity in the whole human population. We can all express up to 6 different alleles for MHC class 1 or HLA class 1 molecule. And importantly for what we study, each of these different HLA alleles will select a different peptide derived from the proteome, carry it to the cell surface for presentation to T cells. So just to reiterate a few of these terms, immunopeptidome is a snapshot of the cellular proteome. It's a very special snapshot because it's quite HLA-centric. As I've said, it's sort of analogous to a shotgun proteomics experiment in the sorts of information it can provide. And obviously we have a cellular-based readout instead of a very expensive mass spectrometer to sort of understand what peptides are being presented on the cell surface. It's influenced by the presence of pathogens and malignancy, and this is obviously key to immune surveillance. The composition of this peptide mixture is highly dependent on the background genetics, and we've just discussed the MHC haplotype effect. It's influenced by the cellular microenvironment, and although I won't have time to discuss it today, inflammation, for example, can change the sorts of peptides that are generated and presented during an immune response. And we've been particularly interested in recent years in understanding novel sources of antigen, or the so-called dark immunopeptidome. So these are peptides that aren't always considered and aren't contained in the reference proteome that many of us would use to search our data against. So things like endogenous retroviral elements. So these are sort of retroviral genes that get inserted into the human genome and can be activated during malignancy. Novel open reading frames that aren't necessarily considered as normal protein genes. And what I'll discuss briefly today is another class of peptides, which are Splice. So these are post-translationally generated in the proteasome. And of course, how do we study the immunopeptidome? Well, I wouldn't be here if we weren't using mass spectrometry to do this. But just to sort of give you a rough idea of what we do, and I'd certainly refer you to this Nature Protocols paper published last year. That should have a 2019 in there. where we sort of really go into great detail into the methodology that we use. But essentially, we take cells or tissues, we isolate the cell surface HLA peptide complexes using immunoprecipitation reaction. We then use an acid treatment to remove the peptides from the cleft of these HLA molecules and dissociate them from the affinity matrix. We separate the peptides and use mass spectrometry in various ways to interrogate the peptide sequences from the cell surface. So when we think about strategies for cancer epitope discovery, this is really where the sort of field is at the moment. So we like to combine genomic and proteomic information to understand the sorts of peptides that are potentially being presented. So we'll start off with tumour tissue or tumour cell lines. We'll isolate the bound peptides and do our immunopeptodomics workflow. We'll use next generation sequencing to understand information about what's being expressed in the tumour, and in particular what somatic mutations may be present. And we'll then combine this information, similar as we do in proteogenomics type approaches, to understand what peptides are there, and combine this with predictive algorithms to predict neoepitopes, which really, I guess, are the flavour of the month at the moment in terms of cancer immunotherapy. And these are peptides that contain a somatic mutation that is specific to the cancer. And from this, ultimately what we'd like to do is come up with an array of vaccine candidates. Now, in the absence of additional information, this approach fails to account for some key classes of antigens. So often we don't search for key PTMs, and there's certainly strong evidence that phosphopeptides, methylated peptides, and deamidated peptides can form potential cancer-specific epitopes. And it fails to account for the class of peptides I'll discuss a little later, the so-called splice peptides that are generated through this proteasomal degradation and then religation of pieces of peptide together to generate untemplated sequences. So the strategy that we take is a little different. We take our tumour tissues and cell lines. We like to treat them with an array of different cytokines and other conditions to really, I guess, recapitulate changes in tumour microenvironment during the development of the tumour. We take this large data set, we do our whole genome sequencing, RNA-seq, etc., to understand what mutations may be present, etc. We do our LC-MS immunopeptodomics workflow, and we sort of really dig deep into the data, and we don't just use the proteogenomic information that's available, but we leverage this particular software package, often the Peak software from Bioinformatics Solutions in Canada, to sort of try and understand the data both in a traditional database type search, but also to leverage the very good de novo sequencing capabilities of this software to find PTM peptides and also splice peptides. And we'll talk about that in the next few slides. And to use this more, I guess, comprehensive approach to come up with vaccine candidates that include other classes of peptides, not just the mutation-bearing neoepitopes. So where are we at with this? And like everything in proteomics, we always like to sort of have a numerical figure. And at least this is what I think we should be getting from the sorts of samples we have in the lab now. So certainly we're quite routinely generating between 50 and 100,000 unique peptide identifications per cell line. Working with tissues and biopsies is a little more challenging, and you'll see some data that speaks to this a little later in the talk. But again, we should be up around 10 to 20,000 peptides from tissues and biopsies if there's a sufficient quantity. And we can use this information for a lot of things. And I'm not really going to discuss these aspects today in the talk, but happy to sort of speak offline to those that are interested. I think you'll be hearing a little bit about PROSIT a little later on today. But there are certainly ways to use these large data sets to do data independent acquisition experiments, for example, on other samples, to improve peptide spectral matches using a variety of predictors. But ultimately what it does do is it allows us to access all classes of tumour antigens. So cancer testis antigens, so these are developmental antigens that are aberrantly expressed, the classical neoepitopes derived from mutations and frame shifts, and the post-translationally modified peptides, including splice peptides. So what are these splice peptide antigens, and why do at least we think they're important? I will preface this with I guess a disclaimer that there is quite some contention in the field at the moment over the prevalence of these antigens, but I think their ability to act as tumour antigens and their existence isn't really disputed. It's how prevalent they are in the immunopeptidome. So there were studies from John Udell and colleagues in 2004, published in Nature, which reported on the first splice peptide in renal cell carcinoma. And this was a peptide from the tumour antigen FGF5, and it resulted from two distinct regions of the protein that were generated in the proteasome and religated in a transpeptidation reaction. And there was a very nice news and views from one of the pioneers of the immunopeptodomics field, Hanjul Ramansu, that I think really sort of described this very nicely, and we'll revisit this cartoon of how it works in a moment. But it wasn't really until 2016 when this paper came out from Michele Misto and his colleagues, involving my good friend Albert Heck, that showed that actually a large proportion of the HLA class 1 bound ligands are potentially best explained as proteasome-generated splice peptides, and this was up to a third of all the peptides. And this was something that I think struck everybody in the field. It created a lot of arguments, but certainly spurred us on to sort of really start to address this and explore the role of the sorts of peptides we could identify using our immunopeptodomics work in the context of cancer immunotherapy. So let's just revisit this cartoon and really consider what the implications of this proteasomal splicing mechanism are. So here we have our antigen of choice represented here. And you know, we understand a lot about this intact protein. We can measure its expression at the transcript level. We know it's genomically templated, so it's almost certainly going to be in our reference proteome or in our proteogenome if it has mutations. And typically what happens in the proteasome, and this is just a machine to break down proteins into their constituent amino acids. And one of the byproducts of that are a lot of different oligopeptides of differing lengths between 2 and 25 amino acids. And some of those peptides are interesting for the immune system and enter into the class 1 pathway, as I've described. What can also occur under certain circumstances is that these short peptide fragments generated during proteolysis can be religated together. And the biochemistry of this is quite well appreciated. There's an acyl intermediate in the catalytic compartment of the proteasome that can undergo nucleophilic attack from the termini of another fragment rather than the typical hydrolysis reaction that occurs to generate this splice peptide through a transpeptidation reaction. And we understand that this can happen from a single antigen, and even more sort of alarmingly, if you like, it can happen when there are two antigens being degraded at the same time, and two regions from different peptides can come together. And we call that the latter trans-splice peptides. And in this example, we call these cis-splice peptides. So these are generated from the same antigen. And we know from the studies from Michele Misto and others that potentially a high proportion of the peptides on the surface of cells are generated through this mechanism. And the challenge is, well, how on earth do you have a reference to actually be able to match spectra generated by MSMS to these untemplated sequences? The approach that Michele and colleagues took was very much to generate very large databases of possibilities of these splice peptides and interrogate them. We thought that was far too computationally challenging and a little fraught with false discovery. So 3 postdocs in my group, Clea Faridi, Chen Li, and Nathan Croft, thought about using de novo sequencing to assist in this process. And the concept here is that we have many high quality spectra that are very difficult to assign for various reasons. And by using de novo sequencing and a combination of library sequencing as well, we're able to identify all the peptides that match to the reference proteome and generate a number of high confidence de novo sequences that can include splice peptides, and then just rank them using a probabilistic function that suggests that most sequences should be linear, that is templated, not undergoing a splice reaction. If not, then they're most likely cis-splice, and can we find an explanation at the cis-splice level? Or if not, then are they trans-spliced? That is multiple proteins, or 2 proteins at least, generating 2 donor segments that can be stitched together. We then research the data to generate a false discovery rate. And obviously we have to be very careful to make sure that we exclude trivial explanations. So we exclude things like endogenous retroviruses, bovine proteins, because that's often a problem in cell culture from the fetal calf serum, and other potential sources of contamination in extended database search. But what we do find is very similar to the science paper in 2016, is that quite a high proportion of the peptides, and it varies between different Hla A and B alleles. These are all human class 1 alleles, and these are all cell lines that only express this one allele to give us very clean data. Again, probably between 25% to 35% of the peptides can be explained by this potential splicing mechanism. So they are highly abundant in the immunopeptidome. But what does that mean if we think about cancer? And I've really sort of abbreviated this aspect of the talk because I want to talk about some quantitative work as well, since that's the theme of today. But we wanted to try and understand, well, what does splicing do to the repertoire of cancer antigen-derived peptides? So we looked at a subset of the splice peptides, and this is data from a patient-derived melanoma. And we looked at splice peptides that had at least 1 donor segment derived from a melanoma-associated antigen. So these are antigens well known to the cancer immunology field that are at least somewhat specific to the cancer relative to normal healthy melanocytes or other types of skin. And, you know, what we find, and I think what is very interesting, is that if we look at a panel of these melanoma-associated antigens, some of these will be familiar to members of the audience, I'm sure. What we're able to show in blue is that we get a number of peptides being presented from these antigens. And if we consider splice peptides derived from these antigens as well, in some cases we see this additive effect where we get additional peptides, and this can only be a good thing if you're looking for targets that are able to recognize and attack the tumor. But more importantly, for other antigens, if we only consider the linear peptides, we don't really see any coverage of those antigens at all. And so it appears that this splicing mechanism may actually improve coverage, improve the sorts of information about tumor antigen expression that has been communicated to the immune system. So we think that, you know, this rather remarkable and quite sloppy housekeeping behavior of the proteasome maybe does have some benefits to the host through this extended proteomic coverage of what's happening in the cancer cell and potential to elicit immune responses to antigens that otherwise, without this mechanism, possibly wouldn't be targeted. So I guess the really important thing is, if we look at these peptides, can we actually see evidence that there's responses to these particular peptides? So In this particular experiment, what we were able to do is go back to patients that expressed Hla A2, a common Caucasian HLA molecule, take a selection of our Splice peptides, stimulate the peripheral blood mononuclear cells of those patients, and show in a patient-specific way that a number of these peptides were able to elicit immune responses. And I guess I'll draw your attention to this particular peptide here. That should read peptide 1, by the way. It's a typo. Sorry about that. But so peptide 6, for example, one of these splice peptides is eliciting a very strong response. So just to orientate you, these are all cells that are expressing CD8. So this is the marker of the killer T cells, and also making TNF alpha, one of the key cytokines that are produced by these T cells. So at least the peptides we find this way are bona fide targets of tumor-specific T cells. So just to sort of summarise this part of the talk, I've shown you where we're sort of at in terms of what we believe is state of the art in terms of the sorts of sort of depth we can get into the immunopeptidome. For the melanoma work, we see at least 5 to 10% of the peptides we can identify are cis-splice peptides. We haven't really explored the trans-splice peptides yet in this system. And that a subset of these are derived from melanoma-associated antigens, and we think the splice peptides that are coming from these antigens are the ones that are really of most interest in cancer immunotherapy. And of course, I showed you that that sort of skyscraper plot, if you like, of the roughly 20% of known antigens that aren't represented by linear peptides, but only represented by splice peptides. And obviously this is closely tied to the patient immunogenetics, but at least for the A2 restricted response, HLA A2 restricted response, what we're able to show is that splice peptides allow further coverage of antigens that don't furbish a suitable peptide otherwise. So what I'd like to move on now is to sort of think about, well, how are we going to translate this? Because deriving cell lines from patients, etc., is great for research, but it's not really going to be that clinically useful. And there certainly are approaches that have been taken. So, for example, Albert Hecht's lab published a nice paper just recently in Nature Communications where they developed organoids from patient tumours. We've worked with patient-derived xenografts, so these are tumour biopsies grown in nude mice. But what ideally we'd like to do with the field is to move from next-generation based prediction of neoepitopes, which is what is sort of being clinically practiced at the moment, to either be able to directly identify or at least validate the presentation of these putative neoepitopes on the surface of biopsies using immunopeptodomics, and not necessarily even having to have this next generation sequencing based approach at all. And of course, the biggest challenge to that is the sample size. So most biopsies we see are sort of around here, which is in cell number terms, if we translate it back to the sort of cell lines we work with, starting to get very challenging to get good depth and to be able to reserve the sorts of clinically actionable peptides we might like to detect. Where ultimately we are going to be is with fine needle biopsies, where we probably only have 1 million cells or so, and maybe only a small percentage of those cells will actually be tumour cells, depending on how the biopsy was taken, the type of tumour, etc. So what we need are techniques that allow us to start to analyse very small samples. And I should say for the cell line data I showed you, we're probably up around here, around 1 to 5 by 10 to the 8, so 100 to 500 million cells to sort of generate enough material to be able to get to our 100,000 peptides identified. So this is work from Sri Ramarathinam, a postdoc in my lab. He's been a long-suffering intern in the lab, and I think he's been with me for 12 years. but he really put his mind to this and has come up with something I think is really exciting. So just to sort of reiterate that when we have enough material, we can generate good quality spectra, and we can use the spectral information as a fingerprint, if you like, to identify a peptide of interest. When we have a low abundance peptide or not enough material to generate enough of the peptide, we get a blurred fingerprint, and this is not enough to identify it. So the approach that Shri came up with was to totally redesign the way we do our immunoprecipitations and to do it in microscale. And this is just a cartoon of how that works. It involves use of these little micro spin columns with different antibodies as the immunoprecipitating matrix. And we can use a series of these microscale columns in series to allow purification of different HLA allotypes or different classes of the HLA. We elute the peptides with acid as usual, use a small molecular weight cutoff filter, so ultrafiltration, to separate out the peptides from the other proteinaceous material. And then we use peptide barcoding, and you just heard from Sanjeeva about TMT, and that's the barcode that we've been using most successfully. And just to sort of reiterate some of what you've heard today, our approach was really to borrow some of the concepts from the single cell proteomics field, and to think about how can we amplify peptide signal from the peptides in our biopsy in a way that allows us to have confident detection. So the great benefit of using isobaric tags is that you amplify the signal of common peptides. And this is represented here in this color code, where maybe these are our biopsy samples here, and we have a carrier peptidome. And the idea is that within the biopsy, there's a small amount of the peptide that in isolation probably is not going to be intense enough either to trigger an MSMS acquisition or intense enough to give a high quality spectra that gives us the identification we want. However, if we have that same peptide represented from a more abundant source in this so-called carrier peptidome here, this will both amplify all of the signals, but it will give us the high quality MSMS to be able to have a confident identification. And then we can go back to this low mass region where all these reporter ions are, to not only get detection, but hopefully quantitation of them. And this is a cartoon of the templex TMT, and often because there's a little bit of contamination between some channels, we won't use the full templex capability, but it is a way to amplify this. And when I describe this to immunologists, I like to call this peptide PCR, because this is essentially what this does to some extent, and I think it's a good analogy for those that aren't familiar with quantitative proteomics. So what does this look like? So I was showing you data from 500 million cells. What if we drop down a lot more? So this is a slightly contrived experiment where we took a B lymphoblastoid cell line and started off with different amounts of starting material. So we had 1,000 cells, 10,000, etc. And just showing you here data from just 1,000 cells using a carrier from the same cell line. So it's sort of the perfect case, if you like, where we have an abundant source of peptides that are relevant, and we've titrated in less and less and less material that has been labelled with a different TMT tag. And what we see very nicely is for the class 1 immunoprecipitated peptides, a typical length distribution for class 1 peptides. For human HLA, 9 mers are by far the most prevalent length of ligands. And here we've also managed to immunoprecipitate the class 2 here, so mostly HLA-DR peptides. And we know that the class 2 bound peptides are somewhat longer. We're able to use a bioinformatics approach to fish out the so-called peptide binding motifs. So each HLA molecule, as I described, has its own preference for different types of peptides. And here you see very nicely the Hla A301 motif, which looks a little like a tryptic peptide, I have to say, but likes this valent position 2 and a lysine or tyrosine at position 9. Similarly, for the B7 peptides here, proline at position 2, leucine at position 9. So these really look like bona fide peptides, and for a particular peptide ligand here, what you see is a very nice dilution, a linear relationship of the intensity as we drop the amount of input material. So again, giving us confidence that this is quite nice. And now at 1% FDR, we're detecting over 900 peptides from an amount of cell input that we would have just dreamt of previously. So what does this look like using a real life sample? So here we took a patient tumour. We were able to generate a PDX from this tumour to generate our carrier. So again, a slightly contrived situation where, you know, we're taking the biopsy and we're making a PDX. So this gives us a large amount of material. And we're able to use that as a carrier and use the biopsy at 2 different amounts, so 19 milligrams of the biopsy and then down to 1 milligram of the biopsy using different TMT tags. Using an exome sequence, we're able to probe both neoepitopes as well as other epitopes presented by this particular melanoma cell line or melanoma biopsy. And again, this is the sorts of results we can see. So here we're only looking at class 1 peptides. The melanoma doesn't express HLA class 2 molecules. And what you can see is, again, this conservation of length. We're getting a good number of peptide identifications from just 1 milligram of biopsy. And this really would have been almost unapproachable without this amplification step. And very importantly, we're able to identify a number of peptides bound to HLA-A2 mostly that are derived from known tumour antigens, and in fact many of these are known T-cell epitopes. And using the quantitative capacity of the TMT lab, we're also able to get some idea of the quantity of these peptides on the biopsy as well. So how are we going to take this forward into the clinic? You know, we'd like to move beyond just using next generation sequencing to predict, to now validate and potentially discover epitopes on the surface of patient-derived biopsies. The carrier is going to be the really important aspect to this, and we're looking at ways to make both generic A2-derived carrier peptidomes and also to use synthetic peptides of known or of known epitopes or peptides derived from next generation sequencing of some of the neoepitopes to probe what's actually being presented. And we've just completed a study, which I don't have time to show you today, where we've taken a whole panel of dissociated cancer cells and interrogated them with this sort of peptide library approach to sort of look at what neoepitopes are being presented. What I did think I'd do just in the last few minutes is to talk about the other way to do this. It's sort of the brute force approach. It's an approach that always appeals to me, and that is, can we find enough money to buy a more sensitive instrument? We've been lucky to work with Bruker to look at the timsTOF Pro, which is proving to be a very nice and very sensitive instrument. And I just thought I'd show you a snippet of data in the last few minutes generated from these guys here. So Shri, you've heard about. Puy has been involved in all the Splice peptide work, and Kirti Pandey, a PhD student at the time, who's now a postdoc in the lab. And you know, we ended up with quite a small biopsy, challenging to look at in any terms. And rather than using the label approach, we thought, well, let's push the system and see what we can see on the timsTOF. And very nice, we were able to show again that we're able to get over 1,500 peptides. They have the right length. This is the mixed motif of the different HLA expressed here, but it makes sense in terms of the sorts of HLA expressed. And we're able to deconvolve these into the various HLA that are bound. And out of that, we're now sort of assessing at what level are we detecting potential actionable peptides within this particular peptidome. We're also able to get very good numbers if we use a normal approach using this instrument, and these are from monoallelic cell lines from different alleles. And again, we're getting very good numbers of peptides, and certainly probably a 2 to 5-fold increase in what we get on other instruments that we have access to. So I'm going to leave it there. Hopefully I haven't gone too far over time. I think I've acknowledged most of the people as I've gone along, members of my lab, collaborators at Monash, and some of our collaborators that we've been working with on various cancer projects, and of course the funding agencies. And I'll leave it there. And hopefully you're all still there.
Great, yes. So wonderful lecture. Thank you, Tony. And I think you kept very much on time as well. So that's great. So to all the participants, I'm sure you are seeing that how proteomics technologies and going beyond peptidomics technology can be so relevant, and many basic concepts which we are trying to convey in the this workshop about peptide sample preparation and the TMT labeling. All of those could be quite valuable despite different type of biological question which you want to address. And these techniques can be very generic to really compare and bring into different type of the questions of your interest, right? So I think Professor Tony, congratulations for this wonderful work done by your team, and really thanks for giving this wonderful lecture. I'll now make it open to the audience for the questions. So let's start with Ayushi first.
Hello. Hello, sir. Hi. This is Ayushi. So I'm a PhD student, and my project is very similar to the work you present here. So my project is also involves the identification of neoantigen using the genome sequencing and the immunopeptidomics approach. So I am actually following your paper, the Nature protocol paper that you presented. I am actually trying to follow that paper. So, so my questions are actually more onto that paper. So first of all, in that paper, you have like for the immunoprecipitation, you have mentioned the immunoaffinity purification approach. So my question is, since the tumor tissue that we got from the hospitals is very less, so following the affinity purification wouldn't be like very impractical, because we get very less tumor tissue and affinity purification needs more lysate. So what is your take, like immunoprecipitation or the affinity purification? Like which approach will be more practical and useful?
So I think we're probably talking about the same thing. So immunoprecipitation, we prefer to do using either magnetic beads or a very small column rather than just beads alone. And this is because we're able to more effectively wash the resin than say just spinning down a little bit of resin in an Eppendorf tube, for example. So I'd always advocate that there's some sort of column-based step. And if you've been following our work, then hopefully quite soon you'll see in some detail how the small scale immunoaffinity purifications we do works. And if you look at that bioarchives paper that I referenced, and I think my slides are available, you'll be able to see some details. And if you want to email me, we can probably tell you a few more secrets.
All right, yeah. So Ayushi, you are looking for more practical things, which I think can be discussed, over the email, or if you have any specific troubleshooting step, we can discuss that with Mr. Tony. Great. All right. Other participants, are there some questions or any comments? Anybody intending to work in this area of peptidomics? All right. Anyone has any questions so far for the session, what we are doing? Okay, Shalini, please go ahead.
Hello. Thank you, sir, for a wonderful talk. It was very interesting. And I just have a doubt. It's not directly related to the cancers, but I am working on one of the parasites where we are trying to do the epitope mapping. So, like in your case, if you're finding any new antigen, I think you can go directly with the samples to find out the related HLAs. But in the case of my study, I'm trying to generate HLAs against the epitope, possible epitopes, which might be there. So I was trying to look into the possible epitopes which can be there, and I had started with the linear part only, linear epitopes. As you mentioned, you have these three sequences of linear, then cis spliced and trans spliced. So we don't know about the cis spliced or trans spliced. But for the linear one only, if we want to go for a HLA mapping or HLA typing, so like you have worked on the bioinformatics part of this as well. So how would you suggest to select for a HLA repertoire to say to start with? Because these are parasite of human only. So what will be a approach to go about this particular thing? If you can guide me a bit on this part.
So in terms of which HLA would be of most interest to study in the context of that parasite, I guess it comes down to the population that the parasite's endemic to. So, in at least working in Australia, we have a large number of Caucasians. Hla A301 is by far the most prevalent allele, so it's present within about 40% of the population. So that's one of the reasons that we look at it. There's also a very pragmatic reason, and that's because we have an excellent antibody that specifically immunoprecipitates HLA-A2. So we're able to isolate that. But I guess in your case, I would look at where the parasite is the problem and what the prevalent HLA types are, the haplotypes in the sort of, I guess, the infected population. Is that answering your question?
So the problem here is that the parasites are very less when we try to extract it, and not much about the proteins is known about that particular parasite. And the issue is that when we are trying to check for the antibodies from the host system, it is cross-reactive with other parasite species. So we thought to first screen it on a bioinformatics level so that we can check for the with minimum resources, because we are already lacking the parasite proteins, and still if we try to screen it against many patients who might have this non-protective antibodies coming up in their body, it will be a very wide range to screen in. So for bioinformatics, we were thinking to screen on the HLA repertoire which are already present on on web databases and all. So
okay, yeah, I mean, so look, I guess if you're looking at what's presented by HLA, often there is an overlap with antibody epitopes, but not always. So I think that you could certainly screen against some of the prevalent class 2 allotypes and see what peptides might be presented. And often that's a clue as to which regions of the protein might also be targeted by antibodies. But these are linear epitopes. Many antibodies recognise discontinuous regions of the protein, and obviously that's not really what splice peptides do, unfortunately. They're a bit different. So I don't think you get information about discontinuous epitopes by looking at the splice peptide repertoire, for example.
Oh, okay. Okay.
All right. So
thank you, sir.
Yeah, thanks, Shalini and Ayushi. And let's thank again Professor Anthony for giving this wonderful lecture and really highlighting that how these powerful proteomics technologies could really be useful to address some really crucial clinical questions which were not possible otherwise. And it also, I think, opens up a lot of potential future collaborative opportunities for many of us here. With that, let me thank Professor Tony for this wonderful lecture and close this session. So thank you very much.